Okay, let's unpack this. Imagine a biological engineering feat so precise that scientists have built a functional piece of the human body,
Right.
but not using 3D printed plastic or silicon or anything like that. We're talking pure human biology, assembled cell by cell. Researchers at the University of Basel have just done this, creating the first fully human-made bone marrow system in a lab. It's basically a blood factory.
And what's so fascinating here is that for decades, this has been, the holy grail of hematology research. We are not talking about just cells floating in a dish.
Yeah.
So our mission today is to unpack the complexity of this breakthrough, which was led by Professor Ivan Martin and Dr. Andres Garcia.
And really understand how they did it.
Exactly.
Yeah.
How they managed to go beyond these simplified cell cultures to build a system that includes the precise mix of stem cells, bone cells, blood vessels, and critically, the nerves and immune cells that define that deep internal environment.
And that's the real shock factor here, isn't it? Because when we think about engineering an organ, we usually think big, hearts, kidneys, that sort of thing.
Yeah.
But bone marrow is so subtle. It's silent. And it's arguably one of the most fundamentally critical systems we have. I mean, it's what gives us life.
Absolutely. And it shows that the most complex engineering challenges are often biological, happening at the micron level. This engineered system, just by mimicking the human environment so closely, gives us a window into blood formation and disease that, well, it simply did not exist before.
Bone marrow is the silent necessity, right? You never think about it. Not until there's a crisis like leukemia, lymphoma, or something like aplastic anemia.
Right.
Which I think brings us to the core issue. Why has it been so difficult for scientists to study bone marrow function and failure in the first place?
Well, historically, scientists have been sort of trapped between two pretty inadequate models. The first one was a heavy reliance on animal studies, particularly mice. Now, mouse models offer a complete living system, but the differences in how blood cells form, what we call hematopoiesis between rodents and humans, they're significant.
So you could spend years developing a promising drug based on a mouse study, only to find out that human physiology just processes it completely differently.
Precisely.
That's a massive roadblock in drug development.
Well, it is a huge one. And the second option was simple two-dimensional cell cultures.
Just a petri dish?
Yeah, you can grow human blood stem cells in a petri dish, but without the physical and biochemical cues of the native environment, the 3D scaffold, the support of cells, those stem cells quickly lose their ability to self-renew or differentiate properly.
They stop behaving like they would inside your body.
They do. They just stop.
So you essentially have to build the stage, the actors, and the audience for the stem cells to believe they are still inside a human bone.
That's a perfect way to put it. And that is exactly where the Basel team stepped in. Their objective was to build a detailed, three-dimensional model made entirely from human cells.
To get past those limitations.
Yes, to move beyond them. They needed a model that could maintain blood formation for weeks, allowing researchers to observe disease progression and test drug responses with just unprecedented accuracy.
So they didn't just try to build the whole bone. It sounds like they targeted a specific, highly active piece of real estate within the marrow.
A strategic decision, yeah.
They focus on the endosteal niche. Why is that specific spot so critical?
Well, the bone marrow isn't some homogeneous soup, It's highly compartmentalized. The endosteal niche is the area right near the bone surface, the inner lining of the bone structure.
And that's where the important stuff happens.
It's the primary residence for hematopoietic stem cells, the mother cells of all blood cell types.
Okay, so why does being near the bone surface matter so much?
It's all about proximity to the structural components, the osteoblasts, which are the bone-forming cells. These cells secrete these critical regulatory factors that maintain stem cell function.
I see.
But more importantly for disease study, this niche is incredibly relevant to blood cancers, especially leukemias. This specific microenvironment is often cited as a major factor in why some blood cancers resist chemotherapy, or why dormant cancer stem cells can evade treatment entirely, which leads to relapse.
So if you're trying to model cancer progression or resistance, you have to model the endosteal niche accurately. There's no way around it.
You have to.
But the complexity you mentioned earlier, the nerves, vessels, the immune cells, that's what pushes this beyond anything that came before. Why are those so essential for a functional niche?
That's the true deep dive into the biology here. Previous models usually missed these regulatory networks. Nerves, for example, might seem a bit counterintuitive in bone marrow, but they play a critical role in regulating hematopoiesis. Adrenal nerves release neurotransmitters that signal the stem cells to move or mobilize out of the niche when the body needs more blood cells quickly.
So without that nervous system input, your model is just static. It's not responsive.
It's static and unresponsive to physiological stress. It becomes a dead model, essentially.
It can produce cells, but it can't respond to the outside world.
Exactly. And the immune cells, specifically the stromal and mesenchymal cells, they are the supportive cast. They create the actual scaffolding and secrete the growth factors. By incorporating all of these fully human cell types, including endothelial cells, to form a vasculature, they're building a living ecosystem where the blood cells are regulated just as they are in your body.
Okay, let's talk blue cut. This is where the engineering specifics come in. How do you even start building a structural scaffold that mimics the inside of a bone?
So they started with the physical foundation. To get that 3D environment, they needed a suitable matrix. They chose hydroxyapatite.
Hydroxyapatite.
Now, this isn't some novel chemical. Hydroxyapatite is the natural mineral component of human bone and teeth.
So it's biocompatible.
Very. Using a synthetic, porous version of this material provided an ideal scaffold structurally similar to the porous trabecular bone you find inside the marrow.
So they're building a tiny, artificial internal skeleton first. But a scaffold is just architecture, right? It needs tenets. How do they stock this mini organ with all those different specialized human cells?
This is where the magic of molecular biology and, precision cellular engineering really comes in. They started with human-induced pluripotent stem cells, or HIPSC.
Right.
So instead of relying on hard-to-source fetal or adult stem cells, they can take simple cells like skin or blood cells from an adult and reprogram them.
Back to that pluripotent blank slate state.
Exactly, back to a blank slate.
I get the blank slate concept. But how do you tell that blank slate to become a specific bone cell and an endothelial cell and a neural cell all at the right time and in the right place? That seems like the most challenging choreography.
That is the core of the breakthrough. It involves applying incredibly specific, sequenced chemical signals like growth factors and cytokines at precise times. The process is called directed differentiation.
Like a recipe.
Think of it like a strict timeline. First, you give signals that nudge the HIPSC population toward a bone lineage. Then you introduce factors that guide a separate population towards forming endothelial cells that will line the internal structure.
And the 3D scaffold itself must play a role here, right? It's not just the chemicals.
Oh, absolutely. The physical environment provides biomechanical cues. For instance, fluid flow through the developing structure, what researchers call shear stress, is crucial for forcing endothelial cells to properly organize into functional blood vessel-like structures.
I see.
The researchers essentially co-cultured these different lineages within the hydroxyapatite scaffold, allowing them to communicate and organize into that complex, vascularized, innervated endosteal niche.
And the result of all that complex choreography, what were the functional specs of this model?
The result was a structurally complete model. It measured about 8 millimeters in diameter and 4 millimeters in thickness.
So about the size of a small pencil eraser.
Roughly, yeah, but just densely packed with functional tissue. And most importantly, this structure was capable of supporting human blood cell production active hematopoiesis for several weeks in the lab.
That extended function is the critical step.
It is. It proves its stability and its viability as a research platform.
And that's what transitions this from a cool experiment into a truly transformative research tool. Which brings us to the implications for medicine and ethics. One of the clearest immediate impacts is the ethical shift, moving away from relying on those historical animal models.
And that ethical drive is central to the University of Basel's commitment, the 3 Rs. Replace, reduce, and refine animal experiments.
Right.
Professor Ivan Martin emphasized this. He noted that while mouse studies have taught us a lot, this new human-based model, and I'm quoting here, brings us closer to the biology of the human organism. It could serve as a complement to many animal experiments.
Complementing animal studies is one thing, but the dream is replacing them entirely, isn't it?
Yeah.
Especially for testing new drugs for blood cancers.
Precisely. If you have a stable, functioning human bone marrow model, you eliminate that translational hurdle we talked about earlier.
Yeah.
You can test the efficacy and the toxicity of new therapeutic compounds directly on human biology, which just drastically streamlines the drug development pathway. This isn't theoretical. This is the immediate application.
However, Dr. Andres Garcia raised a really fascinating engineering caveat that we need to unpack. the miniaturization hurdle. They built this 8mm by 4mm functional model, but he suggested it might actually be too large for practical drug screening. Why would that specific size be a problem?
This gets into the nuts and bolts of pharmaceutical research. When companies screen for new drugs, they don't test one dose of one compound.
No, of course not.
They need to test hundreds, sometimes thousands, of combinations, doses, and exposures all at the same time. This is done using high-throughput screening, which typically involves these plates with 96 wells or 384 wells or even more.
Wait, you mean a single plate has 96 tiny little reaction chambers?
Exactly. And each well is a miniaturized experiment. So if your bone marrow model is 8mm across, you can maybe fit one into a large well, but you can't possibly achieve the density you need for parallel testing.
I see.
To make this a scalable tool for pharma, they need to shrink the model down, maybe to the size of a few hundred microns, so they can run dozens of unique parallel experiments on a single plate. And that saves costly reagents and a lot of time.
So the challenge now isn't proving function. They've done that. It's proving reproducibility and miniaturization. They need to take that complex, functional 8mm city and shrink it into a high-density, reproducible microtown that's suitable for automation.
That is the critical engineering challenge for the next phase.
Yeah.
But if they crack that, and they are actively working on it, the long-term goal of personalized medicine becomes incredibly tangible.
And that is the transformative vision.
Yeah.
Walk us through how this leads to personalized treatments for blood cancers.
Okay, so the ability to generate these functional niches from HIP-BSC means you can take simple cells from a specific patient, say a patient with an aggressive form of leukemia, and create their individual bone marrow model in the lab.
Wow.
This patient's model will contain their unique genetic profile, their specific cellular interactions, and their exact cancer cells all within that niche.
So instead of trying a generalized treatment and just hoping it works, you could test 5 different targeted therapies on the patient's own mini bone marrow system first.
Precisely. You determine which drug or which combination of drugs successfully kills the cancer cells or reverses the blood disorder in that specific, personalized context, and you do it without exposing the patient to ineffective or harmful treatments.
It moves treatment selection from, like, a clinical average to personalized, patient-derived data.
It does. Now, the researchers acknowledge this requires more development, but the fundamental technology to begin this highly customized approach is now proven.
So what does this all mean for you, the listener? We've gone from simplified cell cultures and often misleading animal models to building a complex, engineered human niche that maintains its function-producing human blood cells for weeks. It's a massive step forward for understanding complex diseases like blood cancer and for driving ethical drug discovery.
And this raises a really important question, especially given the accelerating pace of biological engineering. If scientists can now build a functioning, specialized, blood-producing organ niche complete with vessels and regulatory nerves from scratch in a lab, how soon might we see this technology transition from being just a cancer research tool to something more?
Exactly. Could this ability to engineer functional human tissues lead directly to complex regenerative therapies? Could we eventually grow and implant functional, personalized biological components on demand, bypassing traditional organ donation limitations entirely? It suggests we're quickly moving into an era where biology is the ultimate customizable building material,